Mark, in your response to my initial question around processes and applications, you mentioned a few different modalities. You talked a little bit about cytotoxics and radioactives, and you also mentioned cell and gene. We have a few questions from the audience specific to cell and gene, so I think it's probably related to the scale conversation and the modality conversation, probably a good time to ask those. So one of them is, how are single-use technologies being adapted to close? This is from Jonathan, by the way. How are single-use technologies being adapted to close processes at the manufacturing scale for personalized medicines, e.g. autologous cell therapy?
Yeah, that's a great question. Excellent question. It fits in nicely with the single-use discussion. Usually, when you talk about a single-use system, you are also talking about a closed system. So we used to talk about closed, sterile, ready to use. We had all these adjectives, and for many of us who've been in the industry for a while, we've gotten a little sloppy, and we only say single use, and we know what we have in our heads, but we're not being explicit about it. So closed system connections, truly sterile connections, are a very important part of the industry, whether it be tube welding, some of the fancy aseptic connectors, which are made by a whole number of companies. And I'd be remiss if I didn't thank CPC for sponsoring the webinar and making a line of aseptic connectors. They're one, there's many others. Those connections make this work. Those connections put you in a position where you can operate in a lower level clean room environment than you would have to with open aseptic connections. That goes back to our initial conversation about steel versus single use. You need much simpler facilities, much lower energy consumption in them, cheaper to make if you're working with single use. Now in autologous cell therapy, you're dealing with one patient at a time. You're dealing with very small systems, very small volumes of material. And there are a number of aseptic technologies, including the connectors that I mentioned and tube welding to close those systems. What I find fascinating, and we probably won't get into it today, but it'd be fun to talk to the questioner about this one, is what are your requirements as a producer of an autologous cell therapy from a sterility perspective, from a sterility assurance perspective? When you're making big batches of product, like when I was at Merck, we'd make vaccines for hundreds of thousands of people in a batch. You could pull some of that out and test it, and you could do all kinds of things that are directed by regulatory agencies to ensure sterility assurance. When you're talking about one patient, one sample, one dose, or just a handful of doses, it gets a little harder to think about. I know there are some regulatory guidances out there to help us along with that, but I think it's still an open discussion.
Mark, I want to just pull you back to an important point about the vaccines, because when we talk about particularly large virus vaccines that are generally not terminally sterilized by filtration, then I think the lessons learned in your history do speak directly to autologous cell therapy. Again, where process closure, the boundary, the sterility of the final dosage form is established really in a logical progression of each potential risk of point of entry of bioburden into the process and the risk mitigation around that, and really a diligent approach toward process closure. And of course, the regulations don't tell you how to do it, and there's a variety of different methodologies. But I wanted to just pull you back to that to make sure it was explicitly stated that, this process challenge is nothing new. And millions of healthy babies have been, vaccinated with, pertussis or rubella, varicella, all these large virus vaccines that aren't sterilized by filtration.
Yeah, that's an excellent point, Paul. Yeah.
And the bar is pretty high for injecting healthy babies, right?
Yeah, pretty high. Yeah. this child is fine. Let's inject them with something. Yeah, pretty high safety.
Not to make light of any of this.
No,
but
absolutely not.
I think
No.
with autologous, certainly you're generally dealing with very sick patients, and it may be the last resort they have.
Yeah. But from this conversation, certainly there are closed system technologies that if I were working in autologous cell therapy, my company is not an autologous cell therapy company. I haven't worked with those products specifically myself, but I'm familiar with them from reading. I think there are plenty of single-use technologies available to consider, as well as some custom-made equipment that I've seen, these so-called cocoon systems and others that I've seen at conferences. It looks fascinating. But yeah, single-use technology has a lot of options in the toolbox right now.